All right, good afternoon, everyone, and welcome to our third session of Emerald Ash Borer University for spring of 2026. Today, we are hosting Dr. Ann Johnson of the U.S. Forest Service, who's going to be talking to us a little bit on some of the more interesting factors surrounding spotted lanternfly and how they are further kind of adapting or getting adapted by our ecology here. First off, though, I want to make sure that I recognize our team and our partners. My name is Bob Bruner. I'm the Exotic Forest Pest Educator with Purdue University. I am also doing this program in partnership with Michigan State University and The Ohio State University with Sheila Babbitt and Dr. Kayla Perry from those universities, respectively. And I just want to say thank you to both of them for all their hard work. And let's go ahead and get started. And the floor is yours.
Awesome. Hi, everyone. I'm Anne. I'm currently a postdoc here at Michigan State in collaboration with the Forest Service working on emerald ash borer. But today we're actually going to be talking about the research I did for my PhD thesis, which was on spotted lanternflies and how they're using toxin sequestration to basically defend themselves against predators. So, I think I need to do it once and hopefully it'll let me. Okay, so first off, a quick introduction to spotted lanternfly. Spotted lanternfly is an invasive plant hopper that was first found in the United States way back in 2014. Um, and that was in Berks County, Pennsylvania. Since that time, as you can see from this map, it's spread pretty widely, especially along the East Coast, but it's starting to get out here in the Midwest as well. And spotted lanternfly is such a big issue because it actually feeds on a whole bunch of different kinds of plants. It has more than 100 different reported host plants that it'll feed on, but it is especially problematic on woody plants. And in Pennsylvania, where a lot of this damage has already occurred, it was actually a major pest on grape. So, for example, we had one vineyard out there in Pennsylvania back in 2023 that reported that in their 20-year-old vines, they were getting an annual net loss per acre of nearly $9,000 just from extra treatments and things like that. And then on their younger vines, they were actually getting a net loss per acre annually of more than $30,000 because in addition to those treatments, those younger vines, the extra stress from the lanternflies feeding on them made it hard for them to survive over the winter. So they actually needed to replace those vines as well. So I guess just highlights from that spotted lanternfly, major issue on grape. And it's an invasive plant hopper, first found in Pennsylvania, now on the East Coast and in the Midwest. Uh, so, why what we think spotted lanternfly was doing was that we're pretty sure it's using what's called toxin sequestration in order to protect itself from predators. So, sequestration is used by a lot of different animals to achieve a variety of goals, but one of the more famous uses for this is actually for defense. So, a lot of people sort of associate this with monarch butterflies, which you might know need milkweed, and they actually take up and store those toxins from milkweed to make themselves taste bad to predators. And we were pretty sure that spotted lanternfly were doing the same thing with one of their preferred hosts, tree of heaven. This plant is actually an invasive as well. It was introduced way back in the 1700s as an ornamental plant, and it contains a lot of toxins that have also been found in spotted lanternfly, including a group called quasinoids and a group called indole alkaloids. So these spotted lanternflies and the tree of heaven actually co-evolved, which kind of makes us think that they were able to take up and store these compounds from the tree of heaven. Other things that kind of made us think that is that we were seeing these interesting predator-prey interactions in the literature in places where spotted lanternfly had actually already invaded. Um, Prior to reaching here in the US, it had actually already invaded the Koreas and Japan. So research out of there was finding that wild magpies would vomit after feeding on spotted lanternflies, that birds were eating less from little balls of butter that contained ground up spotted lanternfly that had been collected off of that tree of heaven versus ones that had been collected on a different plant that they were feeding on, in this case, Korean willow. And then actually here in the US, we were getting even reports from veterinarians of cats and dogs being brought in for things like drooling, vomiting, and loss of appetite after they had fed on spotted lanternfly. So we kind of wanted to know how the predator-prey interactions here were going to be affected and just kind of confirm that the toxin sequestration is what was maybe causing these. So starting out, in order to figure out how we're affecting our North American predators, we had to know what predators there were. Prior to me starting my PhD thesis, we had a report back in 2016 of wheel bugs, which is pictured here in this slide, as well as a species of predatory stink bug feeding on these guys. And then all the other evidence of predators was pretty much anecdotal, but a lot of people online were saying, oh yeah, my chickens are eating them. So that was kind of what extent we knew before I started my work. So overall, my objectives for my thesis were to identify predators of spotted lanternfly here in North America, determine how the sequestration off of that tree of heaven might affect our interactions with arthropod predators, determine how it would affect interactions with bird predators, and then try and figure out what exactly those actual toxins that were being taken up and stored were. So starting out with identifying those predators, I have actually already published this work. This paper is open access, meaning anyone can read it. You just got to scan this QR code or Google the title, which is down at the bottom of the screen. So the first thing we did was we created a Facebook page back in 2020. It was shared by several different organizations, newspapers, birdwatching groups, master gardener groups, and the end result of that was that by the end of 2022, we actually had nearly 1,300 unique reports of things feeding on spotted lanternfly, 526 of which included pictures or videos of the actual predator, just allowing us to confirm, identifications and behaviors. So, of those predators, slightly over half were arthropod predators, followed by nearly half being bird predators, and then coming in a distant third were our mammal predators. We also had amphibians, fish, and reptiles turning up, but they were in much smaller numbers. Of those arthropod predators, we then further divided them down into order just to see sort of what was going on. The most commonly reported order was Araneae, which are the spiders, followed by Mantidea, the praying mantises, and then coming in third was Hymenoptera, which in our case was wasps and ants, both feeding on our spotted lanternfly. On the bird side of things, the most commonly reported family was Phasianidae. This is the chicken family, and a lot of our reports were chickens, but we also did get turkeys, both wild and domesticated, and peafowl as well, domesticated. So one thing to note about this is, yes, this was the most commonly reported family, but that may not necessarily mean that chickens are like the world's best birds at eating spotted lanternfly. It's just, again, a lot of these things are domesticated, so you have a better chance of actually having people see the chickens feeding on the spotted lanternfly. So there's a little bit of bias there that might sort of push these guys higher than they would normally be. Coming in second was Cardinalidae. This is the cardinal family, and the majority of the birds that we had reported were these northern cardinals, but we actually also had scarlet tanagers and rose-breasted grosbeaks feeding on our spotted lanternfly. And then our third most commonly reported bird family was Mimiidae, and in our case, that was actually all catbirds that we had reported feeding on our spotted lanternfly. We also had a lot of people report to us some additional information, including the life stage, the host plant, and the feeding behaviors. We gave people this graphic here to the right to help identify the life stage, and then we actually compared the interactions between these using some Pearson's chi-squared tests. So, of the feeding behaviors we had reported, we had actually a good number being reported as just eating the spotted lanternflies they saw whole, which is good news. That means that our predators can sort of help control. And a lot of them were actually at least still eating them, removing the wings. But, you know, you don't need, once, if you're eating the rest of the lanternflies, that's still a good method of control. Uh, After that, we were starting to see animals start avoiding eating on these, which in our case was kind of like maybe since we had several reports of change in behavior, maybe them starting to eat them and then like no longer going back to them, even though there were plenty more available, or like looking at them and if there was other food available, preferring that instead. We also, other changes we often saw were things like they would start tearing off the wings, or, you know, they would eat a few and then maybe start spitting them out or dropping them, things like that. Another fun report that we had was actually we did have one case of a human attempt to consume a spotted lanternfly. This was a toddler who attempted to eat the spotted lanternfly and then spat it out, saying it tasted yucky. So there you go. And we did actually get 3 reports of predator death. But again, this is something where it would have been good to have actual, like, a veterinarian or something maybe help identify some of these things. So one of our reports was a frog who reportedly ate spotted lanternfly throughout the season, and then at the end of the season was found dead with no visible injuries. The reporter attributed this to spotted lanternfly poisoning, but there are a number of things that can kill a frog without causing visible injuries. Another person saw a common finch that ate 4 lanternflies and then they saw it die. Again, they attributed this to spotted lanternfly poisoning. This isn't consistent with what we were seeing with the other birds we had reported. So there might have been a different cause there. And then similarly, we had a dog that after eating a spotted lanternfly had a seizure and also passed away. So it's one of those things where we're getting these sort of deaths. They might just be other things causing them that get attributed to spotted lanternfly. In terms of our feeding behavior, by predator type, we found that birds were experiencing illness less frequently than expected. So they were not, vomiting or otherwise getting lethargic quite as often as some of the other groups that we were seeing. Our arthropods were removing wings more frequently than expected, so they were taking the wings off of the lanternflies before they fed on them. Our mammals were actually experiencing illness more frequently than expected, so they were the ones we were really seeing kind of having the vomiting behavior, having sort of those lethargic reactions after eating spotted lanternfly. And mammals were also eating spotted lanternfly whole less frequently than expected. Amphibians died more frequently than expected. Again, it was that one frog. We just had very few amphibian reports. So that one report, kind of seems more significant than maybe it actually is, especially when we consider that all the other amphibians we had reported were eating them whole no problem. In terms of feeding behavior by life stage, our early nymphs were eaten whole more frequently than expected, so things were happy to eat those early nymphs, which were the ones that are first through third instars, still just black with white spots, as were they eating the late nymphs, which for us were just the fourth instars once they get that red coloration more frequently than expected. Our adults were eaten whole less frequently than expected. So it's really the adults was where we were starting to see some of these more interesting feeding behaviors that could indicate maybe that they're not, the predators are experiencing detrimental effects from defenses spotted lanternflies are taken in. And adults were also had their wings removed really commonly. So In terms of feeding behavior by known host plants, our tree of heaven, uh, ones that had spotted lanternfly that had fed on tree of heaven were avoided more frequently than expected and eaten whole less frequently than expected, whereas spotted lanternflies that were observed feeding on host plants other than that tree of heaven, maple, or walnuts were actually observed being eaten whole more frequently than expected, which is again tying back into sort of that idea where this helps show that that tree of heaven is actually the source of those defenses that spotted lanternfly have. Um, in addition, we actually separated out walnut because at the time we knew that spotted lanternflies really liked feeding on walnut as well, and it does produce a lot of toxins, so there was some concern that maybe they would be able to take up and store toxins from that as well, but we weren't really seeing that. The all but two of the ones we had reported feeding on walnut before being preyed upon were actually eaten whole, no problem. So probably not sequestering things from walnut. So conclusions from this study were that a lot of our native natural enemies are feeding on these guys capable of providing some measure of control. The removal of the wings is similar to behaviors that we've seen predators do in monarch butterflies. In monarchs, a lot of those toxins are actually stored in those wings. So by removing them, you can get rid of some of them. We might have a similar sort of thing going on here. And then removing Tree of Heaven could help remove some of these defenses against natural enemies. Removal of Tree of Heaven was already being used in spotted lanternfly management. So just getting rid of them, they're already an invasive plant, might be helpful. And tree removal seems like a good option because black walnut, which is native, doesn't really seem to be a concern. This can also help us start to understand the effects on the ecosystem of the spotted lanternfly invasion. Our next objective was to determine how sequestration affects those interactions with those arthropod predators, since they were our most reported group. This is again a published paper, also open access, so all you need to do is scan this QR code and you should be able to read it if you are interested in getting all the nitty-gritty details. But what we did here was we had 10 different predator species for some choice tests and 9 for no choice tests. The 9 were 2 lacewing species, 2 ladybug species, rabid wolf spiders, Chinese praying mantises, Carolina mantises, spine soldier bugs, and wheel bugs. And then the 10 were those again, plus we added in black carpenter ants. We provided the predators with a spotted lanternfly that was raised with access to Tree of Heaven or without access to Tree of Heaven in those no choice tests, and then one of each in those choice tests. For the ants, things had to be a little bit different because the ants actually needed to stay with their colony in order to go foraging. That's why they only did the choice test, where we actually gave them 10 of each kind of spotted lanternfly and basically saw which ones they took away more of. And then we went and we checked for predation at 24 hours, 48 hours, and 1 week, and compared the proportion of each that were preyed upon using Fisher's exact tests. And the end result is that in our no choice tests, and actually in our choice tests, we did not see any significant differences in our tests. Our ladybugs and lacewings did not eat any of the provided spotted lanternfly at all, so they're not really good predators of these guys. But everyone else was happy to eat all of the spotted lanternflies provided to them in the no choice test. They would eat them pretty equally. And even in the choice tests, sometimes we had cases where they ate one that had access to Tree of Heaven first, and they were still willing to go back and eat the other one. So this seemed to indicate to us that the arthropod predators, at least the ones that we tested, weren't really being deterred by those stored defenses that spotted lanternflies were taking in, which could help us use them then later to actually control these populations. So to kind of see which of the commercially available or easily collected predators that are out there might help us in the future with control for these guys, we did an experiment seeing which ones could actually reduce the populations of spotted lanternfly the best on grapes that were set up in these little pop-up enclosures. So what we did is we set up pop-ups with a grape plant and a tree of heaven plant in them. For the nymphal stages, we put in 25 spotted lanternflies each, and then for the adult and spotted lanternflies in the egg masses, there were 10 in each cage. And we used commercially available or easily collectible arthropod predators. We tested our green lacewings and our 2 ladybug species against egg masses, first instar and second instar spotted lanternflies. And then we tested our 2 mantis species, our wheelbugs and our spine soldier bugs, against all of the nymphal instars and the adults. We had 3 reps for each predator at each life stage, plus 3 controls, where it was just a pop-up with those populations and no predators in them. And we monitored them for a week, and then compared them using a repeated measures ANOVA, followed by a Tukey post-hoc test. So, our eggs, we did try those egg masses, like I mentioned, again, using those lacewings and ladybugs. You'll discover that I continually tried to use lacewings and ladybugs, and they just were not eating any life stage for me. So they did not actually eat those eggs either. Then, for the first instars, we found that both Chinese mantises and spine soldier bugs were eating more spotted lanternflies than the control, where we had no predator in that enclosure. Whereas something like you'll see my green lacewings and my multicolored Asian ladybug up here actually did worse than the control, but we're not significantly different. So for our other life stages, it was quite similar. For our second and third instars and our adults, the Carolina mantises, the Chinese mantises, and the spine soldier bugs actually all reduced those spotted lanternfly population levels significantly relative to the control. And for our fourth instars, actually, all of the predators we tested, those spine soldier bugs, those Chinese mantises, those Carolina mantises, wheel bugs, everyone did a good job reducing those populations relative to the control. Now, I will make the note that my wheel bug here maybe didn't get a fair shake of things, because what we did is, for the commercially available predators, we applied them at the rate that the seller recommended. And then for the ones that we field collected, of which wheel bugs were one, we just applied it at the same rate as the most closely related commercially available predator. So the most closely related commercially available predator we had to wheel bugs were our spine soldier bugs. And what's really cool about these guys is that they're actually what's called a communal feeder. So they'll work together to kind of bring down prey and feed on it. Whereas wheel bugs tend to be a lot more territorial, and they will definitely feed on each other. So what we might have been having with the wheel bug is that in the other life stages, they definitely were feeding on each other. So they might have actually kind of fed more on each other than the actual lanternflies we had for them. Until the fourth instars, they were just able to bring down those populations too. So what was our conclusions? Those spine soldier bugs were consistently bringing down those spotted lanternfly populations the fastest, giving it pretty good promise as a potential control method for spotted lanternfly. You can actually see here, these are some spine soldier bug nymphs all working together to bring down that spotted lanternfly. But before we actually use them as a control method, we'll want to see if they will feed more on spotted lanternflies as more become available, either directly or by having more offspring. How they'll interact if there's other prey available. Again, these were in enclosures. The only thing they could feed on were each other or the spotted lanternflies. So it makes them really easy for them to choose the spotted lanternflies. And how would they actually do out in the field? A lot of times predators maybe will leave the area. The good news for spine soldier bugs is that they actually have what's called an aggregation pheromone, meaning they like to be near each other, and they can draw each other in with this chemical pheromone that they can smell. So we could put some of that out in our field with our spine soldier bug to keep them there, but it does still need to be tested. So our next objective was to see how it affected that other large group of predators we had, the birds. And this actually is in the same paper as the one where I went over what toxins are actually present in spotted lanternfly, which can be found here. Again, either scan the QR code, it's open access, or Google the title. You should be able to find it pretty easily. Okay, so the first thing we did for the birds was we wanted to test those earlier life stages of spotted lanternfly. So we would freeze spotted lanternfly nymphs at those various nymphal instars and place 10 frozen and then, frozen to killed and then they thawed, along with 10 live mealworms into cups on top of house wren nest box around campus at Penn State, where I was at the time. Those live mealworms were actually just to sort of draw the attention of the house wren because if there wasn't movement, they wouldn't necessarily know that there was an insect there for them to feed on. But once they found that cup with those live mealworms, they would happily switch to the dead spotted lanternflies as well. Those spotted lanternflies were raised either with access to that tree of heaven or without access to it. And we would just observe to see what the birds actually did with those spotted lanternflies, just to make sure that they were actually like eating it themselves or feeding it to those chicks, as opposed to, say, just like tossing it out of the cup and dropping it. We would then go back. We would do those observations either ourselves with binoculars or with trail cameras, and then go back after 24 hours to see how many spotted lanternfly were actually missing, and then compared the proportion gone between those that had fed on Tree of Heaven and those that had not. So for our nymphs, we found that the birds would prefer to prey on the ones that had not had access to Tree of Heaven for all of the different life stages. This was actually surprising to us because we had that paper that I referenced in the beginning where the birds were avoiding butterballs with those spotted lanternflies that had fed on Tree of Heaven, but not the ones that had fed on Willow. They had actually tried it with third instars as well, and they didn't find a significant difference. And then again, with our community science project, we had people reporting them eating the earlier life stages, no problem, eating them whole. Whereas here, where they had a more concentrated difference, the birds seemed to be able to tell. So what we think might sort of be causing that is that for that paper in particular that I referenced, and even in our observed field things, we don't really know what those spotted lanternfly were feeding on before they were feeding on the host plant they were observed on in the experiment. So if they had fed on Tree of Heaven and then moved over to Willow or moved over to Black Walnut or something, they might have gotten fed on whole by the birds, and they might not have had that much of a difference because they'll still have some of these toxins taken up and stored from previously feeding on that Tree of Heaven. Whereas ours, we know for certain that the ones we raised to never feed on Tree of Heaven had never fed on Tree of Heaven. So there might have just been a bit more of a difference. We also might have had a bit of a difference because our birds were nesting, and that might have made them more sensitive. So in order to see how adult spotted lanternflies would be treated by our North American birds, we actually created 2 different batches of suet. One contained spotted lanternfly that had fed on Tree of Heaven, one contained spotted lanternfly that did not. We set them up in these double-sided feeders that were just kind of free swinging, and put out our trail cameras to capture videos of the visiting birds, and then compared the number of times that the birds would peck each type of suet. And we did find that birds pecked the suet made with spotted lanternfly that had not fed on Tree of Heaven significantly more than the suet made with spotted lanternflies that had fed on Tree of Heaven. It's a bit hard to tell with this graphic just because we found that our woodpeckers, even on the see what they preferred or didn't prefer, would just peck so many more times than like some of our little songbirds. So we also divided these out. These were the 5 most common species of small birds. We actually found uh, a significantly higher average number of times than chickadees and white-breasted nuthatches that they would peck at that suet made with spotted lanternfly that had not fed on Tree of Heaven. The other ones, they were still higher, just not significantly so. And then for woodpeckers, we found that both downy woodpeckers and red-bellied woodpeckers were again pecking that suet made with spotted lanternfly that had not had access to Tree of Heaven significantly more. And hairy woodpeckers, it was more on average, just not significantly so. So, what are our conclusions? Again, our birds seem to be affected by those sequestered defenses throughout the spotted lanternfly life cycle. That was different from our hypothesis. I didn't mention this one. Yes, we also had different species of birds that could play a role in it as well. Our reared non versus the field collected. So we knew ours had never fed on Tree of Heaven as opposed to field collected. They might have previously, and that sensitivity of nesting birds. We actually had an undergrad come in and do a little project with us where we actually saw what those birds, we kind of looked, would those birds feed on the spotted lanternfly we provided to them, or would they give them to their chicks? And you'll see that these white ones blocks are when they fed them to their chicks, and they would only feed spotted lanternfly that had been reared without access to Tree of Heaven to the chicks. They would still eat spotted lanternfly that had been reared with access to Tree of Heaven, but they wouldn't give it to the chicks. So they might have just been a bit more sensitive since they were nesting or wanted higher quality prey for their offspring. This does indicate to us that, again, that removal of Tree of Heaven could help encourage bird predation, because if they can tell when a spotted lanternfly is fed on Tree of Heaven and then it hasn't, then hopefully they'll eat a bit more of them. And then that final objective, identify and quantify which toxins were actually being sequestered by spotted lanternflies. To do this, we got a whole bunch of spotted lanternflies. We reared them out, both with access to Tree of Heaven and without. And then we also field collected some. And we basically ground up these samples and we prepared them with what's called the catcher's method, where basically you run them through a cleaning process to sort of clean up all of those other things that might be in a spotted lanternfly before running them through what's called liquid chromatography mass spectroscopy to actually identify and quantify those quasinoids, in our case, that were present, those toxins that were present. We also dissected our spotted lanternflies to try and identify where in the body spotted lanternflies might be storing these compounds. So we had the integument, which is like your exoskeleton of an insect, your fat bodies, which are these little clusters of fat, the ovaries, which is where the eggs are, the gut, and then salivary glands, where the saliva is produced. And we also tested our host plants just to make sure that we weren't accidentally, on the ones we were rearing, introducing any quasinoids that they could take up and sequester at all of these different life stages. So the quasinoids that we were able to test for were ilanthone, which is one that's very associated with Tree of Heaven, glucurobenone, 2-acetylglucurobenone, 13,18-dihydroglucurobenone, grandolactone A, quasin, and then neoquasin, because these are ones that we actually had purified samples of. So for those plants that we reared them on, we were happy to find that only our Tree of Heaven was containing these quasinoids. I, in this diagram, have just grouped ilanthone and then all the other quasinoids together down lower there because we found, and this is true for all of them, even though I'm just going to show you a summary of total quasinoids, that ilanthone is the majority of the quasinoids present in all of these. Grandolactone A was the other one that kind of like made up a good portion. We were not really seeing neoquasin at all, but for the most part, most of what was making up that total quasinoid concentration is going to be ilanthone. So for our total quasinoid concentration in our adult spotted lanternflies versus the phloem from the trees, we were seeing higher concentrations in spotted lanternfly, both ones that we field collected, ones we had reared on the greenhouse on Ailanthus, and actually even ones that we had not given access to Ailanthus, then we were finding in the phloem of that tree of heaven, both in the field and in the greenhouse. When we divided out dissected our spotted lanternflies, these were field collected ones. We found that those highest concentrations were in the salivary gland and the gut, but we were also finding them in the cuticle, in the ovary, pretty much everywhere within those spotted lanternflies. And then across their life cycle, we were kind of have this like little swoop here you can see where they actually did still have them in the eggs that they were laying, except for the ones that we gave no access to Tree of Heaven. So those eggs are at the end because what we did is we used eggs we had field collected and then reared throughout that life cycle. So this white line here, no Ailanthus, that means eggs and then no further Ailanthus. But again, we were finding those quasinoids in the ovaries. We seem to be finding them in the eggs. It's starting to look like those adults might actually pass down some of those quasinoids as well. So that might also contribute to why our firsts and seconds seem to be a bit more defended than like our thirds and fourths. And then adults, when they're feeding heavily again and really like being on that tree of heaven, they're kind of gaining those defenses back. So from those analyses, we did determine that spotted lanternfly are capable of sequestering quasinoids from Tree of Heaven. Ailanthone, again, being that main one, with grandolactone A making up most of the rest, but there were little bits of pretty much everything except neoquasin in there. The concentrations we found were highest in the gut and salivary glands. That's kind of what's first interacting with that phloem, so maybe it just kind of gets taken up there pretty highly. But they were also very much so present in the ovaries and the cuticle. The ovaries, again, might allow those toxins to be passed on to the eggs and help defend those, whereas the cuticle might help explain some of that wing removal behavior we were seeing, because those wings are pretty much all just cuticle. So if you remove them, then you can decrease your toxin load. We also found that the quasinoid levels in our greenhouse reared Tree of Heaven and spotted lanternflies were more variable, which is just an interesting thing to note if you're ever trying to do experiments with spotted lanternfly in the future. If you're greenhouse rearing them, you might have a bit of an effect there with what you actually get out, as opposed to something that has just sort of occurred in the field. So, conclusions, yes, we do indeed have generalist predators here in the United States that are feeding on spotted lanternfly. Yes, spotted lanternfly is capable of sequestering quasinoids from Tree of Heaven. Yes, it is affecting some predator-prey interactions. In specific, our birds really seem to be affected. They were able to tell throughout that life cycle. Our arthropods really weren't, but that's still good news because then we have our arthropods who can maybe help with control a bit more. And then mammals, that third most common group, we didn't really specifically have a study looking at it, but we did have some mammalian visitors to our double-sided suet feeders in that bird study, which you can see a raccoon here, and it was an incredible difference. Almost no mammals were willing to feed on that suet containing spotted lanternfly that had fed on Tree of Heaven at all. They pretty much always fed on that suet that was, had no access to that Tree of Heaven. That might be mitigated or might be caused by the scent, because even I could actually smell the difference between the two. The one where they had been reared on Tree of Heaven very much had Tree of Heaven has a very distinctive scent. A lot of people describe it as rotten peanut butter. And even I could smell that in the suet that was made with those spotted lanternflies that had fed on Tree of Heaven. So I would suspect that those mammals could also smell that difference and were probably avoiding that suet and giving that preference to the one where they had been reared without access to Tree of Heaven. But that would need further study. This also gives us good hope that our native natural enemies could provide some measure of control. Those spine soldier bugs are native. I thought they seemed like they were doing really well, but encouraging them with things like what's called conservation biological control, where we help improve the habitat for them, or even birds for that, removing the Tree of Heaven would kind of fall under that, or augmentation biocontrol, where since those spine soldier bugs are commercially available, and have that aggregation pheromone, you can actually draw extra ones into your population. They might be helpful in that way in the future after we do more testing. And then, personally, I would be really interested in seeing how toxin sequestration affects parasitoids and pathogens of spotted lanternfly. That's kind of a dream thing for the future, just because we do know in other species that like these stored toxins can help maybe avoid parasitism or help recover from illness. So that would be fun to look into as well. Which, thanks to all of these people, so many people, very helpful in getting this project done. We had funding provided to us by USDA NIFA, by Northeast SARE, more USDA NIFA, Pennsylvania Department of Agriculture. So all of these awesome people who, without whom this work would not have gotten done. So if we have any questions, I'm sure there's some in the chat. These are also my emails. The top one is my Michigan State one. It's the one that gets checked most often. So if you don't want to like type out a question here or ask it, that's the one that's easiest to reach me at. I occasionally still check these other two, but definitely if you need to reach me, try the top one. And I will look at the chat, I think. Yeah, there we go.
All right, we
Aha.
did have one question in there. Someone was asking what chemical was causing the difference in behavior, and you kind of answered that, and they saw that too when you were referring to the secondary compounds in Ailanthus. Do we have any additional questions, though? Just to give people a little bit of time if they are writing out questions. Sheila put in the chat that our next webinar coming up for Emerald Ash Borer University, A Tale of Two Fungi, Biological Control Options for Tree of Heaven Autumn Olives, being given by Carrie Fearer on May 14th at 1 p.m. Oh, hey, we got one. Cool.
Here we go.
So, do you think these results suggest that SLF populations will be most prevalent on Tree of Heaven and be otherwise mostly controlled by predators?
So for me, I so one thing we noticed just when we were learning how to rear out these guys is they actually really like to be able to move between different species of plants. They don't really start heavily going onto Tree of Heaven until those uh, red fourth instar late nymphs and then the adults. Before that, even like the very little ones don't really feed on trees at all because they can't get through that bark yet. So they're mostly on like herbaceous plants, or like if they're up in the trees, they're up in the leaf, feeding on the leaf veins. They really like when we were rearing them in the greenhouse. They really like the tender shoots, the little viney shoots off of our grapes, or the runners on our strawberries. Um, so I think that having Tree of Heaven in your area is definitely going to help them. In addition to my work showing that it can help deter predators, we've also found that it can actually help speed their development, and that might just be because Tree of Heaven produces a lot of sap, so they can get a lot of food. And if it's speeding development, they have a bit more time before wintertime freezes come to kill them off to actually like lay more eggs. So you'd probably still get higher populations where Tree of Heaven is, but I think they're going to be just fine in areas without it. Um, so yeah. That's why you should go to the next webinar on how to control Tree of Heaven.
Do we have any additional questions? Oh, here we go. So they're not species dependent, or are they dependent on Tree of Heaven?
They are not species dependent. They can achieve maturity and reproduce all without access to Tree of Heaven. So, yeah, they like having it, they don't need it. Yeah.
I'd like to follow that up with my own question. And did you see which species they would prefer to go ahead and reproduce on, or did they show any preference at all?
We did do a study with that. I'm trying to remember because I was not the one running it. Um, I know that if Tree of Heaven is there and they are in those later stages, Tree of Heaven was definitely the preferred thing. I'm pretty sure it was maples that came in next, but don't quote me on that.
That
um,
kind of matches up with what we've seen here at Indiana, is that they tend to gravitate heavily towards maple after spending some time on Tree of Heaven at the start of seasons.
yeah.
So I would love to see some studies run here in Indiana that reflect that.
Yeah, yeah. I know like maples were a big one. Cherries were a big one. Um, river birch. I know we did river birch and they liked it. Walnut, of course, since I even made a point of mentioning that one. Um, they really liked walnut, especially as fourth instars. Not so much for reproduction, but still interesting to note. Um, yeah, so they like all sorts of things. And again, those little ones like viney things. So grapes, oriental bittersweet, we were seeing them on, poison ivy, which, yeah, so
All right. Well, thank you very much. I'm not seeing any. Oh, we got another question, of course. Have they been shown to kill bittersweet?
I've not seen any evidence of that. No. But even like, I mean, a lot of the damage they do is more so weakening trees and making them susceptible to dying from other things. So, um, you know, even the grapes they killed, it was usually they weakened the grapes and then it was actually stress from the winter that killed them off. Or like when they kill Tree of Heaven, it's usually like young and otherwise stressed trees. So it's things like that where they're more of a contributing factor to killing off plants than actual like a direct um, plant killer. So yeah, we don't really get bittersweet control out of it, unfortunately.
Okay. Do we have any other questions for Anne? Okay. I am not seeing any. So I will just say if you do have additional questions, feel free to send them to me, or Anne shared her contact information as well. Other than that, thank you everybody for coming, and I hope we get to see you at the next one on May 14th talking about Tree of Heaven control. Everybody have a great week and stay safe out there.